Welcome everyone. This is day 2 of lecture 1 in general microbiology. And so we're going to continue on from yesterday's slide. Our last meeting, we ended on the comparison of microorganisms and their various attributes. And we'll be filling this in together during our next Zoom meeting. So please work on that. And then when we meet during Zoom, we'll compare notes and we'll see whether you guys have the right answers. And so we're going to move forward from there and look at the 3-domain system. The 3-domain system was devised by a scientist named Carl Woese. And Carl Woese, back in 1978, was studying the genetic contents of various life forms on Earth. So all the different life forms were compared with respect to their ribosomal RNA content. So the little r is in reference to ribosomal. And this refers to ribosomes. Some people have had basic biology, or if you remember back to a biology class, when you learn about how cells work, the ribosome is the part of the cell that makes proteins. And because all life forms need to be able to make their proteins, this is one of the most important central functions of all life forms. And so ribosomal RNA is part of how ribosomes are built, is that their structure contains this genetic material, RNA. RNA comes from DNA, and so Carl Woese was looking at the genetic sequences of this RNA that helps to build the ribosome. Ribosomes are built partly of the ribosomal RNA, and then also partly with proteins, ribosomal proteins. So looking at the ribosomal RNA, it became apparent that there were 3 major domains or groups of life forms that had very similar ribosomal RNA. And so bacteria are known to have very similar ribosomal RNA that they use to make their ribosomes and then make their proteins from that. And when Mr. Woese was looking at eukarya, the eukarya are, oh, I'm sorry, I skipped over archaea. When Mr. Woese was looking at archaea, archaea were known to be a lot like bacteria, but at the genetic level, their ribosomal RNA was significantly different. So they got their own domain as well. And then finally, the 3rd domain are the eukarya. And eukarya are all of the nucleated cells, right? They have a nucleus and they house their DNA there, but when we look at their ribosomal RNA, we can see, like Woese did, that they all have very similar protein-producing genetic elements. And so when we compare the 3-domain system to the old-fashioned 5-kingdom system, if you've heard of the 5 kingdoms, about 10 years earlier, in 1969, there was Robert Whittaker, who came up with Whittaker's 5-kingdom system. And so both the 5-kingdom system and the 3-domain system can coexist. And I may just take a moment here to then show you how the 5-kingdom system fits in. So if you've heard about the 5 kingdoms, the 5 kingdoms includes plants, or plantae, kingdom plantae, animals, which would be the kingdom Animalia, and then fungi get their own kingdom in Whittaker's 5-kingdom system, and protists. So that includes things that we've heard a little bit about, like algae and protozoa so far. And along with the slime molds, this makes up the kingdom Protista. And back in 1969, when Whittaker had devised the 5-kingdom system, we have 1, 2, 3, 4 kingdoms here. The 5th kingdom were what he thought were basically all bacteria. So earlier in history, the archaea were grouped with the bacteria because they're so much like bacteria. They used to be called archaebacteria. So the bacteria and archaea were the fifth kingdom. And the fifth kingdom was referred to as Monera. Monera. So you had your 5 kingdoms that made up Robert Whittaker's 5 kingdoms. But we're going to go ahead and focus on the Carl Woese's 3 domain system, and not worry as much about the 5 kingdoms that Robert Whittaker came up with. All right, here is a visual of a 3-domain genetic tree. Sometimes they're called phylogenetic trees, as we get very specific about the different life forms and where they belong genetically as life has evolved. And if we look at this figure more closely, we can see that the first forms of life appear down here, and fossil records and scientists who study evolutionary biology have taken the evidence and considered that life is estimated to have started as long ago as 3.58 billion years ago. That's the number that most books and scientists agree on. That life began about 3.5 billion years ago. I have read articles and publications that show that maybe life started even as late as 4.1 billion years ago. But I'm going to go with the numbers that we see in our textbook that we're using, and that's the number that you hear most often by biologists. So we'll stick with that. If you're not sure how that is in relationship to the age of the Earth, geologists and scientists who've studied Earth history believe that the Earth was established as a planet around 4.5 billion years ago. So there could have been roughly 1 billion years of Earth's existence without life on it. And no one really knows how life made its way to Earth, maybe on an asteroid or a comet or something that crashed into Earth, and life may have been carried from somewhere else. We're never really going to know the mystery of that. That's part of cosmology. And so we'll skip over that part, right? Did a lightning bolt strike Earth and suddenly life appeared, or the hand of God or something? We're really going to just have to not worry about that. But we do know that life is here and we're part of it. And the earliest life forms, way billions of years before our appearance in the record of life appearing on Earth, we have an ancestral cell line. Most likely these first ancestral cell lines are bacteria. Bacteria-like. We know that bacteria are the most ancient form of life. And bacteria are still to this day the most diverse. We have various different groups of bacteria that we'll be covering in this course. And so here we have all these bacteria. Separately, we can see that at some point archaea had grown off of a branch that goes away from bacteria, genetically speaking. And I find this to be a fascinating figure, primarily because we see archaea branching off in the direction that we, the eukaryotes, or eukarya, had come. And that's strange if you think about it, because that means that the domain archaea have ribosomal RNA that genetically relates more similarly to us, the eukarya. Nobody knows why, but at some point, additionally, we can see that the eukaryotes, which means we have a nucleus, started about 2 billion years ago. So this little stretch from 3.5 billion years ago to this little branch right here represents over 1 billion years worth of time. And this is a very rough timeline. It's not super precise, but somewhere in this range, the eukarya appeared about 2 billion years ago. Some books, I think our book may say 1.8 billion years ago, but I'll be rough on this and just say about 2 billion. And so we have this timeline of history of life on Earth, and it's according to Wose's domain system. All right. Speaking of history, I would like to spend quite a bit of time, a fair bit of time, on the history of microbiology, because I think it's important that as a student of a course like micro, that you guys understand why this particular subject exists. It's not a very old science compared to other types of science where people have been studying stars and astronomy for a long time. Numbers and physics, mathematics, all these different science-related fields are thousands of years old, yet we're only a few centuries old. And we're going to see here that a few key players gave rise to microbiology as we know it today. So I want you to know about those famous scientists and how what they did brought us to this classroom, brought us to you guys needing to know quite a bit about these invisible life forms. And it all really started back when the invention of the microscope occurred. Before the microscope, there was no real way to prove that bacteria and other invisible life forms could exist. And so the story starts just before microscopes were invented. We include Robert Hooke first, and Robert Hooke was reporting that with a magnifying glass, so that he had almost like a microscope, but just a really good magnifying glass. He took thin slices of cork from the bark of a tree, and he held it up to the light, and he looked with the magnifying glass and saw that in the cells of the bark of the tree, there were these little boxes. And back in his time, in 1665, there wasn't the name cells. In fact, he coined that term by referring to cells as little boxes. And so we had the beginnings of this idea that what we are as life forms is made up of the smallest units of life called cells. So we're huge lumps of cells, right? We're multi-organismal, but each organ in our body has a complex number of specialized cells. And if you go below that level, then you're talking about organelles, and below that level we can just talk about chemicals. So really the smallest unit of life are these cells, and not long, just a handful of years, or a couple of years really, of history, we had Antony van Leeuwenhoek, who had invented the first microscope. And so Leeuwenhoek was credited with having observed the first bacteria, invisible life forms that he referred to as animalcules. And so it wasn't understood that bacteria were a thing. The word didn't even exist, so he called them animalcules. And the way that Leeuwenhoek's microscopes worked was that he had fashioned them to be held up to the light, like a little metal plate. And on the end of that plate, or in front of that plate, he had a little mounting pin where he could put drops of water or some kind of specimen that could contain life forms in it, bacteria in this case, that he observed first. And Leeuwenhoek didn't set out to be known for this. He was a Dutch linen merchant, so he was selling various types of threaded linen products, and so people would come in and say, hey, I'd like to buy a tapestry or some sheets or whatever, tablecloth. And if you've ever shopped for linens, you would probably hear about thread count, right? Even to this day, if you go to the department store and you buy a linen, how you shop for the quality of that in part is how many threads are in there. And the more threads, the heavier duty, the more durable, and the better it might launder, etc. And so Leeuwenhoek was really good at grinding glass lenses. He wanted to take the magnifying glasses that he could make and show his customers close up, hey, look at the quality of these linens. Look at how well they're put together. And so he would give them a close-up look. But then after a while, he got bored of looking at linens and thread count. And so he got very good at making tiny little glass lenses that he crafted in such a way that if you held them up to the light and you had a small sample of something to look at, that you could inspect it very closely, microscopically. And one day, as the story goes, Leeuwenhoek had found some rainwater samples out behind his shop, or something in which, if you see how water, when it sits around, can start to grow stuff. He had looked, and maybe he saw mosquito larvae or something like that swimming around that had seemingly appeared out of nowhere. And he wanted to take a closer look at that, and so he took a drop of water, put it onto his microscope that he had fashioned, and to his astonishment, he didn't just see mosquito larvae or other bug larvae close up. What he was able to observe, and that he made drawings of, were these little tiny bacteria. And he described every major shape and type of bacteria, size of bacteria. So here he shows a rod-shaped organism, and here he drew that some bacteria he observed had the ability to move around and swim. He was able to observe little tiny cocci, which are spherical shaped bacteria, large rods, bent rods, curved rods, even curlicue bacteria, which are referred to as spirochetes. So he made these recordings, and he was really excited to tell other people about it. At the time, one of the most important organizations out there was the Royal Society. The Royal Society in London during the 16 and 1700s, if they recognized news and brought it to the people, that was one of the biggest ways of being officially on record for having done something significant. So he took his recordings and reports, and he made a couple hundred microscopes, and he sent them around to anybody who would listen, that he figured was important and would pay attention. And the Royal Society in London had said, well, we're not so sure about this Leeuwenhoek stuff, and nobody really, maybe they didn't know exactly how to use this microscope well enough to understand what Leeuwenhoek was talking about, and he wasn't initially recognized for this groundbreaking work that ushered in this new subject, this new science called microbiology. So it took a while, and some of Leeuwenhoek's creations are still around. There are original microscopes that are in museums that Leeuwenhoek made, and the one that I would love to go see someday is in France. The Pasteur Institute is a place where when you walk in, they do have research that goes on in the building, but there's also the museum portion of it. So tourists can come and take a look at original piece of equipment like Leeuwenhoek's microscope. And we're going to see other things that are in the Pasteur Institute as well. And so that's part of the very beginnings of microbiology as a history. Also going on during that time, there was a big debate about, could life come from a spontaneous event? This was referred to as spontaneous generation. And the idea was that if you mix together the right ingredients for life, maybe the right amount of air and food and water, that poof, something could appear like bacteria by just this event of combining the right elements. And that would be considered common sense. Even to this day, there are many people from cultures around the world who believe that if you just simply add the right elements together, life will appear, it will spontaneously generate. So in essence, we can capture this by just simply remembering it as life can begin from non-living elements. So we can say life from non-life, or non-living. If you take these elements and together they form something that now is magically living, and that's why we have to fill in the words like magic, because no one really knows how that would happen. But again, it comes from a common sense perspective about what's observed. Microscopically, you might not notice that there are bacteria coming into the picture because they can float on air currents. They can already be present in the water and in the items that are non-living. And so we can't see those come into play. And there were scientists during this time period. Now they could track bacteria. They could look for invisible life forms that would get into systems. And some of these early microbiologists had come up with the idea of biogenesis, which was defied common sense because you would need a microscope to prove that biogenesis is the more likely or the accepted explanation now in the scientific community. And it says that living organisms come from pre-existing life forms, that life doesn't just spontaneously generate out of nowhere. And that still doesn't address how life got started on Earth. But if you're going to be a practicing medical professional, you have to think, if we can sterilize medical equipment and gloves and an entire surgical theater, including filtering the air, then we could remove all life. And that's a good thing when you're trying to prevent surgical infections, post-surgical complications, because maybe there was dirt and dust and germs that could get into things. And so biogenesis is addressing this idea that we can remove all life, and if we ensure that life forms, even invisible ones, don't get back into that system, that sterility can be achieved. And that biogenesis is how we operate from understanding contaminating events. All right, so in essence, we're saying that biogenesis requires that life only appears from previously existing life. Life can only come from life itself. And this is a very important understanding that we have to operate from. And you'll see as we go through this course that this is the standpoint. And we could joke about this to a certain extent, right? It would be like saying, well, I don't know how so-and-so got pregnant. Life just spontaneously occurred, right? I can't imagine what would have brought about, somebody getting pregnant. I mean, there would be an example, right? We make the connection between pregnancy occurring because invisible sperm and egg had fused, and those don't get there by accident or spontaneously. And that would be an example of how biogenesis is the way that we need to think about these events, not it magically happened, which is a spontaneous standpoint. Okay, now this particular argument between spontaneous generation and biogenesis started centuries ago with an Italian physician, Francesco Redi. And Francesco Redi had said, so long ago, he said, maggots do not spontaneously generate from meat. And here he was fighting common sense. And this is one of the first stories that we know of that brings about proving that spontaneous generation, or disproving spontaneous generation, and bringing us to the conclusion that biogenesis was at work in his experiment. And here's how it worked. He basically just took some raw meat, stuck it in a jar, and the meat was sealed in the jar so as to prevent flies from coming in. So what would you predict would happen here? Right? Common sense today, you know that fly larvae called maggots would be prevented from growing on meat that's sealed in a jar. He was not looking for bacteria. This was not part of his experiment. Bacteria weren't obvious, and it was before the microscope. But by simply preventing the flies from getting to the meat, no maggots. Okay. Now, he did this experiment, and he was a good scientific thinker, right? He had a control. And the control was that flies could get into the open jar that he had right alongside the sealed jar. And even if he didn't know exactly that there were fly eggs that were being laid on there, invisible pieces of life that eventually hatched and started growing the maggots, but his experiment showed then the maggots appeared, as you would imagine, because those invisible fly eggs deposited by the flies landing inside of the open jar of meat, this is what would happen. So anyway, this was a pretty big deal at the time, and of course there were critics, right? So-called antagonists, people who said, well, come on, Reddy, you can't just put a jar on there and seal it shut, because there's no way for maggots to appear to spontaneously generate if there's no air. And so, they were telling him that you're preventing life from occurring because you've sealed out the air. And so he said, well, we could do this experiment one more time. And he had a clever way of disproving these critics by putting a little netting on there. And so fresh air could get through, but the flies couldn't get in. And what do you think was the result? No maggots, even though there was fresh air. So this was a giant story that still lives on to this day because of the clever way in which he did his experiments. So was Francesco Redi a supporter of spontaneous generation or biogenesis? Think about it. We'll talk about it during our Zoom meeting. But that's something to go back and review, right? What did his experiments prove? All right, but he wasn't the only scientist during this time period and forward that wanted to address this particular topic. John Needham. This was a man that was famous in England, and quite a bit later, 1745 versus the 1600s. So mid-1700s, Needham had tried to do some experiments with broth. And broth can grow bacteria very easily. All you have to do to make some broth is take any kind of nutrient source and boil it for a long time. You want chicken broth, put in some chicken parts and boil it for a long time, and the soup or the liquid that comes out of there would be chicken broth. And so John Needham had made some kind of broth, and if he left it around after cooking it, if he left it long enough, it would spoil. And the spoilage on the broth was caused most likely by bacteria. All right, now the bacteria in the spoiled broth would appear cloudy, and we'll talk about how when a broth turns cloudy, that's called a turbid liquid. And so his broth spoils were turning turbid or cloudy, and he was basically saying that after boiling it, it should have actually sterilized the broth. His idea was that boiling the broth would destroy any life forms, and that's sensible. But then by leaving the broth around, which Needham, I guess, wasn't real careful about it. He let this cool off, and after leaving it around for long enough, boom, he had spoilage. Bacteria grew. Okay? And so Needham kind of set back this argument about biogenesis because in his view, the bacteria grew out of nowhere. And so, you know, this was a guy who really was saying that, hey, look, I'm showing that spontaneous generation is still at work. All right, but from where did the microorganisms come from? Where did the microbes appear? And if you read about it in your book, and in this case, I'll direct you to the answer, there probably was some opening to the fresh air. In fact, I had read some recounts that Needham's experiments after boiling, he had taken clean flasks and dumped the boiled broth into those. Or if he had a pot of boiling broth and then he left that open to cool down to the open air, you can imagine that bacteria could fall into his system at some point before he just left it there. Basically, bacteria can be carried on air currents. So can fungal spores and such. The air that he let into his experiment was a source of contamination. And because Needham was somebody who was revered as a great scientist of his time, people listened to his reasoning and saw his experiments that he had explained, and they believed that spontaneous generation was still very much at play in our environment. Okay. 1745 was Needham. By the way, you don't have to memorize exact years. I'm just trying to give you guys a timeline so that you can effectively think a little bit about how these stories unfolded over centuries. In fact, it was over 100 years later that finally somebody came along, a man by the name of Louis Pasteur. And Pasteur is arguably the greatest microbiologist of all time. Pasteur, as you may think of him through the word pasteurization, had come up with a really unique design in which he did a modified experiment that Needham had performed. He took broth and put it into a flask, and then he did a little flame work on this very unique flask that he made where he bent the upper neck of the flask into an S shape. It's sometimes called a swan-necked flask or an S-neck flask. And this is the key to Pasteur's experiment. In his experimental design, he boiled the broth so that he could essentially sterilize it. Through extended boiling, he was fortunate enough to sterilize it. Now that's something that involved a little bit of good fortune or luck, if you want to refer to it that way. And the reason is because we now know that there are some forms of bacteria called endospores or spores. And endospores can be in samples, could have been in his liquid broth, and if they had been in there, which they most often come from soil. So if he had dirt in his experiments from the soil, this boiling process may not have been successful. But fortunately, when he made his broths, there were no endospores that could survive boiling. And so what happened was the boiling did its job of sterilizing, and then he let that cool down. All the while, his experiment was left open to fresh air, and even after the experiment, he left it open to fresh air. Surprisingly, to most people's shock, after he used this experimental setup, no contamination, no bacteria grew. So we have to think about why, all right? But before we do that, let's figure it out then. If no bacteria grew after boiling and boiling, and we know that this food source was full of nutrients, it was a type of nutrient broth, then why didn't the bacteria grow? So even after cooling it down, okay? So it says here, furthermore, that Pasteur's experiments, after he was done with them, he'd made multiple experimental setups. He had broken the neck off of one so that it no longer had the S neck shape attached to it. And when he broke off that part of the flask, there was bacterial growth. So I'd like you guys to think about why. In this case, why? Okay? And then he also had one where instead of breaking off the neck of the flask, he went ahead and tipped the flask so that some of the broth that had remained sterile for however long he wanted it to remain sterile went into the neck of the flask. But before it leaked out, he tipped it back up very carefully, quickly enough to keep the liquid back in the flask. And in this case, there was growth as well. So in both of these events, there was bacterial growth. Let me rewrite that. Bacterial growth. And remember, he knew that because he could see the cloudy spoilage. And you could put it he could put a sample under the microscope, and he would see that there were bacteria causing that cloudiness to occur. Okay? So in either case, let's think about why. I'm going to leave that to a discussion during our Zoom meetings, and let's make sure that next meeting we have a chance to address this topic a little further. Okay? One last thought about this before we move on. Pasteur is celebrated and recognized as having basically set to rest the argument for spontaneous generation. He disproved spontaneous generation to a level that people were happy with his experiments. He was a hero as a scientist, and we'll see why over time. But at the Pasteur Institute, this is another one of the things that's in the display case. I'd love to see an original Leeuwenhoek microscope, but I'd also love to see one of these setups. Pasteur had made a bunch of these, and this particular S-neck flask is on display with a date on it that basically says sterile since, and then years worth of time, or however long they've kept that there. As long as this remains undisturbed, bacteria will not get into that system, and it can, even though it is open to the fresh air, this setup remains effectively sterile. And so that's why Pasteur is also credited with having established what we call aseptic technique. Aseptic technique is this conscious way of operating that you're preventing germs from getting into your systems, or whatever it is that you're doing, whether it's surgical aseptic technique or aseptic technique in the microbiology lab, or in some cases food, right? You're trying to, package food so that it won't spoil, and the better you are at keeping things aseptic and without contamination, therefore you then have something that hopefully won't spoil or won't get infected. Okay, so here's a question that asks us to review this particular topic, and I'll save this for our Zoom meeting next time, moving forward. There was an age called the Golden Age of Microbiology, and really for this period of time when Pasteur and his greatest contemporary scientist that he worked with off and on throughout his life was Robert Koch. And Koch was in Germany, Pasteur was a French scientist, and so they had had a chance to cross paths and work together on many experiments. But these two gentlemen together are sort of the original fathers of microbiology. And so this is the discoveries and the techniques that they developed brought us to the modern form of what we see in this subject today, microbiology. And here is a reference taken from your book, a timeline of all these great discoveries by microbiologists and biologists during different parts of microbiology. This particular figure shows the Golden Age, when there were rapid numbers of discoveries that were earth-shattering in terms of establishing our whole worldview of what bacteria are, and viruses, and other life forms as well that are pathogens in most cases. And it highlights some of the greatest discoveries, most of which were Nobel Prize winning. So when we hear about the Nobel Prize in scientific endeavors, most of these scientists are recognized at the highest honor. I do not require you guys to know all of these. If you're studying for the next quiz or exam, we're looking at having to know the names of the scientists that I specifically highlighted, the experiment that those scientists contributed something important from, and then, well, that's about it. So you don't need to know where the scientist is from, like the country that they're from, and you don't need to know the years or the dates. But nonetheless, I think it's still important that we know a bit about the greats. So continuing on with Pasteur as the greatest, at least in my opinion, I think he's the greatest. I said he was a hero, and that's because in France, he was credited with having saved the French wine industry. So we all know how important wine can be in various cultures. The French love their wine, and every other bottle of wine back in the 1800s would basically become spoiled. And so when you go to a restaurant and you order a glass of wine, and the waiter or server comes over and opens the bottle, shows you the bottle and everything, and then they pour a little sip for you to see if the wine is acceptable. It's not so much that you're there to basically tell them, oh, I don't like that variety. It's to drink the wine just to make sure that it hasn't spoiled. If wine has spoiled, it turns sour, it'll taste maybe even like vinegar, because some of the spoilage bacteria that get into the wine will actually turn the grape juice sugar to vinegar. And so anyhow, Pasteur was trying to address this very frustrating event where French wineries were losing, the vintners were losing half of their product, and Pasteur had said, okay, well, when I look at the spoiled wine, when we put some of that spoiled wine under the microscope, we can see the overgrowth of bacteria. And he had figured out that the wine that wasn't spoiled didn't have those spoilage bacteria, and that there was at least a good presence of a good number of yeasts. And so part of what he discovered was that yeasts could brew the wine properly, but there were bad bacteria that he wanted to get rid of, and so the whole development of pasteurization came about. He figured out that if you gently heat the wine or alcoholic beverage, beer, whatever it would be, for long enough, then you would end up having a situation where you could kill potential spoilage bacteria. So the original formula for pasteurization was a gentle heating. And he figured out that the minimum gentle heating was 63 degrees Celsius for about half an hour. And that would be enough elevated temperature so as to remove the potential spoilage bacteria. And then even furthermore, Pasteur then decided that the yeasts that he would commonly find on grape skin that was naturally present, that he could grow some of the yeasts separately and then put those yeasts into the wine so that when it was bottled, it would have a really good chance then of not spoiling. So this was the process known as pasteurization, and then he followed it up with yeasts after the gentle heating. And you might ask, well, wait a minute, why not heat hotter than 63 degrees Celsius? And Pasteur tried that. Undoubtedly, he tried boiling at 100 degrees Celsius. But the problem with that is then, how's your wine going to be? Think about alcohol, right? Alcohol evaporates, and boiling would evaporate the alcohol. And I don't think that would be probably what people would want. So thinking about it, he had to find a way to keep the alcohol in the product that was fermented. But also, if you think about boiling, it could change the flavor of things. And in fact, pasteurized products do have a slightly different flavor than unpasteurized products. So unpasteurized beer, for instance, if you drink some beer that's out of a bottle, it's almost always pasteurized. And that pasteurized beer doesn't taste quite as good as its unpasteurized counterpart. Go to a bar and order the same beer that you've had in a bottle, but if you can find it on tap, it almost always tastes better on tap. And that's because pasteurization is not performed on keg beer. You know, if you've got this giant keg of beer for a party and everybody's going to drink it like overnight, or you're at a bar where there's mass consumption, then you don't need the pasteurization and the beer actually tastes better. Anyway, moving forward, guys, another major player around this time period is we need to take time and understand the contribution of Ignaz Semmelweis. Semmelweis is credited with understanding that hand washing was super important. He knew about germ theory. He knew that there were invisible bacteria. He was a physician who had observed that in various medical facilities that pregnant women were delivering babies, and that there was a terrible, sometimes deadly type of infection that was going on associated with doctors who were delivering the babies and having the babies come down with something called a childbirth fever, all right? And septicemia, which was the major symptom of childbirth fever, refers to the fact that dangerous bacteria, streptococcal bacteria from down in the birth canal, right? So ladies can have this undesirable pathogenic type of streptococcus. It's called group B strep, or even group A strep. And we'll talk about group A and group B streps that can potentially get inside the baby during the first few moments of life. And what happens is that, or what was happening was that the doctors who were delivering the babies were not washing their hands in between patients. And so they had these birthing wards where there might be, like, Semmelweis in his reports had said that he had observed whole rooms full of ladies, like say 4 ladies all giving birth at various times, but kind of at the same time. And what would happen is the doctor would deliver a baby. We know how messy labor and delivery can be. The hands could be all, bloody. There can be mucus and tissue fluids and such, and some of these invisible bad bacteria. And then the obstetrician would go from delivering that baby and just maybe wiping the hands off with a rag, or who knows, and then right on to the next delivery. And what was happening was germs were being transferred, and there were incidence rates of childbirth fever which had a high rate of mortality, to where as many as more than 1/3 of babies were coming down with this infection, and then maybe dying as a result. And when Semmelweis observed this, he told those doctors, hey, you really need to wash your hands. And I know that's common sense today to think that we should wash our hands, but we still battle with it, don't we? So you may hear reports that doctors in hospitals are not washing their hands enough. You can tell people, hey, you need to wash your hands after every patient, or you need to wear gloves, but you need to change your gloves. And it's easier said than done. We still have to keep in mind the lessons that Semmelweis brought about, because when hand washing was implemented, he was able to reduce those rates of childbirth fever down to 1%, and that's an incredible drop by just simply washing hands. And there were people that were offended by him. Fellow doctors who said, oh, come on, Semmelweis, you can't tell me how to do my job. You're not me, and I know that I know how to do my job. And there's ego there, right? A lot of people have that ego because you can get to a very high level of training, but they weren't thinking about germ theory. And we still have to have that on our minds as if you're a medical professional. It's something that can never leave your mind. And if you make mistakes, your patient's life could be at stake. And so Semmelweis is someone we should always remember for that contribution. How about good old Joseph Lister? Everybody's probably heard of the product Listerine, and it comes in honor of the contributions from Lister so long ago. So back in the 1800s, there was, just like there was a Semmelweis, that there was a Lister who was very aware of germ theory, and he was a surgeon in England who would operate on his patients and make sure that everything was as clean as he could possibly make it. He knew that just simply washing your hands with soap and water might not be enough. That in fact, not all bacteria are killed by soap and water and simply washing hands. So the surgical equipment, the surgical theater, including the gurneys or the operating tables, the air, everything that he could possibly think of, he would wipe down or mist with something that he referred to as carbolic acid. Carbolic acid is now known as phenol, all right? And phenol is very much an antibacterial substance, a disinfectant. And so he was the first scientist, actually doctor, who could operate on his patients and not necessarily have them die afterwards of, say, post-surgical infection. So if you were an important person in England at the time, if you had the money, if you had the access, you wanted to go to Lister, because if you were going to have to go under the knife, here was a guy that might not kill you as a result, either during or probably more likely after, due to infections, complications. And so we should keep this man in mind as well, right? This is somebody who influences today the modern surgical techniques. The idea that we need to kill all bacteria when possible in these surgical settings. And so let's continue on. Let's look at contributions by Robert Koch. All right. So I was saying that Koch worked with Pasteur, and he also did a lot of work on his own. And Koch is probably most famous for the postulates. Koch's postulates were broken down by we break it down in most books by 4 particular criteria that Koch came up with to prove the germ theory of disease. The germ theory of disease is to say that when we come into contact with certain invisible life forms, mostly bacteria, like he was working with in this setting, he used bacteria that cause anthrax. When we come into contact with these organisms, then there's a chance of infection. And so there's this correlation, there's this actual connection between exposure to a germ and then the disease process that can result from that. And even to this day, we have people who debate about these kinds of things. You say, oh, how could this tiny little invisible thing that we can't barely prove that it exists except for under the microscope, how could that possibly be getting into our bodies and causing some serious infection, let alone something, a deadly event, a deadly disease. And so this deadly disease was being studied in cows. Cows and other farm animals even humans, we can pick up anthrax, and these potentially deadly bacteria get underneath the skin, and it's called cutaneous anthrax. Cows form these lesions on their hides where the big sores on the cows were being studied by Robert Koch, who basically scraped up some of a sample, took a clinical sample and spread it onto a Petri plate, and then he grew the bacteria. He'd isolated the bacteria that were causing the deadly infection. And so here's a picture of how Koch's postulates can be achieved. Now, instead of cows, this picture shows mice. And so if you wanted to perform Koch's postulates, you could say, all right, let's find a deadly disease, or let's find some disease process. Doesn't have to be a deadly one. But you find this and you do an autopsy, or at least some clinical sampling from this diseased animal. And so let's say for a moment that the dead animal here in the picture has a dangerous bacterium like Klebsiella pneumoniae. All right, we'll hear more about that in the coming lab sessions. And so Klebsiella pneumoniae, a deadly cause of pneumonia, and then this mouse maybe has gone septic and the bacteria have got into the blood. You could draw blood, take a little sample of that blood and smear it or spread it around onto a Petri plate and grow the colonies of bacteria. Blood normally would not have bacteria growing in it, so that would be a good sample to take, where if the subject was healthy, the blood should not grow bacteria, but then if you take a blood sample and you spread it around on a plate and it starts growing bacteria, especially a whole lot of colonies of the same kind of bacteria, you've probably isolated the pathogen. So that was number one on Koch's postulates, was to find that the pathogen is present in all cases of the disease. So pick your disease. Koch had been studying cows, and all the cows that got sick and died with the symptoms of anthrax, he was able to do an autopsy or some sort of sampling, as I was describing, and then seeing that the pathogen keeps popping up in all of these cases. Now, the second of Koch's postulates, second step would be that you did isolate those pathogens. So the second Koch's postulate is the pathogen must be isolated in pure culture. Pure culture means that no other contaminating bacteria or other life forms are present in the plate. So if it's bacteria that are responsible, you get a plate of just pure, say, Klebsiella pneumoniae, or in Koch's case, he was studying Bacillus anthracis. That's the genus and species name of the anthrax-causing bacteria. So whatever organism it is, you isolate it in pure culture. All right, step 3. What are you going to do to test to make sure that you have a bacterium or whatever pathogen you choose? How are you going to prove that it causes disease? And this is sad. I'm glad as a scientist that I never had to work on live animals, that I never had to do something like this. But this is a very important type of experiment where you take a healthy, susceptible lab animal and you expose them. The word for this would be inoculate, all right? So Koch would have to take a needle or a sharp object like a scalpel and pick up some of the bacteria and put it into the cow's hide, get it under the cow's skin to start up an infection, or in this case, someone could inject the mouse, and then so that's step 3. Okay, step 3 is challenge. That means to expose a healthy, susceptible animal. Okay, so if you challenge a healthy, susceptible animal, what's going to be next? Now, ask yourselves, it's a little bit of a common sense question. Is the animal going to drop dead, like right after you inject them? Probably not. In fact, no, not from the cause of this disease, right? There's an incubation period. There's a time that it takes for the pathogen to get into the body of the subject that you're challenging and to multiply and grow in that lab animal. And then what do you think the 4th step is? It's, again, kind of common sense. If you think about it, you're just going to wait to see if symptoms of disease develop. And you have to then bring this connection full circle, which is to do another autopsy or clinical sampling and look for the cause of those organisms. All right, so the 4th and final step of Koch's postulates is to see the symptoms of disease, accompanied meaning along with the organism. So the pathogen has to be re-isolated into pure culture again. We need to see that same organism that was initially isolated from the previously diseased animal or dead animal, and so we bring this full circle. And those are Koch's postulates, all 4 of them. And to this day, Koch's postulates remain as the gold standard for proving the germ theory of disease, making that connection. And there's no rule that says that Koch's postulates have to be done with bacteria. The same process could be done with viruses. This same process could be done with fungi, fill in the blank. Whatever potential pathogen, microbiological organism, we could try to perform all 4 of Koch's postulates. Now, this brings up a couple of key questions. These are critical thinking questions that I would like you guys to think about now, and then during our next Zoom meeting, I would like to walk through them with you. So the questions would be, how many of Koch's postulates would be difficult to meet? What could we have difficulty with meeting the first of Koch's postulates? And I want you to think about yes or no and why. The same could be true, could be said about step number 2. Could it be difficult to perform step number 2 or 3 or 4? And so I want you guys to think about that during our discussion during Zoom. Okay? We're almost done with today's lecture. Just a couple of remaining slides. We have vaccination. What a topic. Guys, we're going to spend more time on vaccinations, but the origin of vaccinations in modern medical science, at least in the West, this is somewhat turning a blind eye towards some of the stories that come out of China and parts of the East in Eastern medicine. But nevertheless, in modern medicine, as we know it in Western culture, we recognize that vaccination and the name vaccination is born out of Edward Jenner's contribution. So just before the 1800s, in 1796, Edward Jenner was a scientist who had made a an observation. He was aware, and it was understood at the time that farms would sometimes experience exposure to smallpox, just like it would outbreak in the cities, and smallpox was a dreaded illness where many people would get sick at a high rate of mortality, and it was dreadful. I mean, like if smallpox came to your neighborhood, it was time to pack up and get out of town if you could. No one wanted to die of such a terrible disease where all these welts called pox would develop all over the body. Fever, spiking fever would result, and people would die as this virus would just take over the body. And so when smallpox would make its way out to, say, the rural areas like a farm, what would happen as a result sometimes would be that the people who milked the cows, so milkmaids, ladies whose jobs were to wake up and milk the cows every morning, they were in close contact with cows that would sometimes experience a disease called cowpox. And if these ladies had, just through close contact on a regular basis with cows, experience exposure to cowpox, then when smallpox would come around, they would be the ones that somehow would either get a mild infection or no infection at all with being exposed to smallpox. And so Jenner made this connection that somehow these ladies would be saved from the deadly battle with smallpox. And knowing that, he decided that the way to develop a medical treatment was to take a cowpox lesion where there was a sore in the animal's hide, and take scrapings from it and mix that up into a little bit of liquid that could be injected. Or in the original version, what he did was what was called variolation. Variolation is where you take scrapings from, say, the lesion of a cow, and you spread it along like a needle or a small scalpel. And the way that he did this was he took the upper arm of whichever person he wanted to vaccinate, as it eventually became called, and he put this in there underneath their skin. And this would develop a small but not deadly infection. And the reason is because cowpox may be lethal to cows, but humans are less susceptible to cowpox virus. And the cowpox virus was similar enough to the smallpox virus that when the person recovered from this vaccination event called variolation, their immune system would build up defense towards cowpox and also smallpox. And in fact, this was the first purposeful immunization. And by the way, that's what vaccination, the name came from, which is the Spanish word for cow. So vaca, which is the Spanish word for cow, relates to vaccination as a word. Now, it's been said, for just an extra moment, Jenner first tried his vaccination on a young boy. If I remember correctly, the boy was about 10 years old, and he was vaccinated and later reportedly had come into contact with people who had smallpox, and he did not get the disease, and he survived exposure to smallpox, which was observed as the vaccination being, that was the first evidence of the vaccination working. But Jenner didn't necessarily know everything there was to know about how the vaccination worked. He had the right idea about immunity, but he didn't really make the connection as well as Pasteur. Pasteur knew about vaccination, and he was in a much later time period in the story of microbiology and medical treatment and such. And Pasteur is credited with figuring out how vaccinations do what they do. How do they actually work? And the way that he made this discovery was with working on a vaccine for cholera. And so Pasteur would take cholera-causing bacteria. I'll write down the name of these. They're called Vibrio cholerae. And Vibrio cholerae are pathogenic bacteria that when animals, as well as humans, when we swallow this organism, it will bring about a fatal diarrhea. And that sounds kind of wild. I can't imagine just dying of diarrhea, but it does happen, you guys. As odd as that sounds, what happens is these bacteria colonize the gut and they turn on the waterworks, so to speak. These bacteria cause the gastrointestinal cells to malfunction so that these GI tract cells start pouring out water. So tissue fluid and eventually just the very water that makes up most of our bodies, right? You've probably heard that most of our body is made of water. Imagine losing so much of it through diarrhea that these bacteria make it so that you can't survive. You die basically due to dehydration. And it's kind of wild because when people get cholera, they get set up in what's called a cholera bed. Maybe you've never heard of these. I'm going to take a little trip to the internet, so let's just sidestep this for a moment since we're on the subject of cholera, and then we'll come right back to finishing up about the vaccine that Pasteur was working on. So I'm going to put here cholera bed into a Google search. Check that out. You can see that a cholera bed might not be much more than just a makeshift cot with a hole in the middle of it and a bucket underneath. So some of these are adjustable. You might be able to, make it more like a hospital bed where a person can sit up a little bit, but they're not going anywhere. They would barely be able to make it to the bathroom, and as messy as diarrhea is, the bucket's going to collect maybe up to 18 liters a day. If you can imagine that, losing, a 2-liter bottle of soda is this big. Now multiply that by 10, and that's why it can be deadly, at least in the very beginning, short term. If you have such a disease, this is the reality. And by the way, the diarrhea is filled with all those infectious bacteria, so it's very contagious. All it takes is for someone to put that into their mouth, even in visible amounts, right? Not washing your hands, not being careful. Even if you are careful, this is a really contagious situation. So the dreaded cholera was being worked on by none other than Louis Pasteur. Now Louis Pasteur, he was dosing lab animals. So he took a rabbit and he fed Vibrio cholerae that he isolated from someone who, maybe from someone's infectious diarrhea, and he grew it on Petri plates and he fed that to the lab animals, or he could even inject that to the lab animals, and these lab animals would develop the symptoms, and then he would watch the disease process and eventually do an autopsy after they died as a result. And as evil as that sounds, to have to perform all 4 of Koch's postulates to understand how to make a vaccine, it was really Pasteur's way of trying to find a way to possibly make it so that people could escape this deadly consequence, this deadly disease. So during this period of time, he figured out that some of the bacteria growing in the Petri plates had lost the ability to cause disease, which is bizarre. So imagine sometime during these experiments, Pasteur took a plate of Vibrio cholerae and he fed it to a lab animal, and nothing happened. So the lab animal, the rabbit, seemingly, was for sure exposed, but then didn't have disease. And so then Pasteur was a little frustrated with that or baffled by it, and so he took another healthy animal, another rabbit, and fed it Vibrio cholerae. Nothing happened. So he decided that something maybe had gone wrong with the plate of bacteria that he was using, and so he went out and he got a fresh isolate, searched for cholera going on somewhere, got some of that infectious bacteria, and then went back to the lab. And he took that animal that had survived the first exposure and fed them the fresh isolate of Vibrio cholerae. Miraculously, the rabbit survived. Strange. Then he took that other rabbit that had survived, and the same thing happened. The rabbit survived. So then he went ahead and he took another rabbit that he had never used. So he's got the fresh isolate of Vibrio cholerae pathogen. He's got a fresh rabbit that's never been part of the experiments. Exposed that animal, boom, the animal died. And so what he was able to conclude out of this was that if he was able to use a source of Vibrio cholerae that had become non-pathogenic or less disease-causing, so maybe it had mutated somehow and lost the ability, it allowed these rabbits' immune systems to build strength and some form of immunity so that they then would be able to survive the pathogenic fresh strain of Vibrio cholerae. And this is how vaccinations were discovered to actually work. So this is the essence of vaccines, guys. You take a part or a piece or a weakened pathogen and you mix it up into usually an injectable, and you put that into the person's body. This hopefully doesn't give them infection, let alone kill them. The person then, their immune system will respond, and you'll end up with a person who hopefully is effectively immunized, so that if they ever do run into the full strength pathogen that should cause disease, they won't actually develop disease as a result. And by the way, this brings up a fascinating topic about vaccinations, which is true of all forms of security, so to speak. If you want to immunize yourself or someone against the disease, you do this vaccination, but yet the news is no news, right? The news is that nothing happens because silently, invisibly, the person's immune system is doing the job that it's supposed to do. And when it comes to security, no one really talks about non-events, right? So we get into this false sense that, hey, we haven't seen that forever. It must not be around anymore. But that's when vaccinations are being given. As soon as we stop giving vaccinations, and we have people traveling from all parts of the world and mixing, inevitably, people getting on jet planes and carrying diseases from one country to another, from one area to another. The next thing you know, without the vaccination, these diseases come roaring back. We can see this with measles. We're seeing this with polio. The measles episode happened at Disneyland just a couple of years ago. Polio is on the comeback in Pakistan, of all places, because people are worried about immunizations. Either it could be against someone's belief system, or they're worried about the safety. It happened with MMR vaccines. Falsely, someone thought that MMR was a connection to autism, and we now know that that scientist was found to be criminally responsible for facilitating bad information that killed a lot of people as a result. And still to this day, we have people denying vaccination because they're worried about safety. And it's a complex subject that we'll come back to at least a couple more times this semester. All right, this is our final slide for today, and it has to do with antibiotics. Between vaccines and maybe antibiotics even more so, this is one of the greatest life-saving inventions humankind has ever come up with. And it started almost 100 years ago. In 1928, Alexander Fleming was a scientist who was studying staph bacteria. So he was doing work as a microbiologist, taking samples from people's skin, staph, we now know, has deadly infections, right? Antibiotic-resistant staph, like if you've heard of MRSA, methicillin-resistant Staph aureus. 100 years ago, MRSA was not a problem, right? Antibiotics hadn't been invented, but this was the advent of that discovery that was still today is a very powerful treatment against various bacteria, including staph infections that are responsive to antibiotics. And what he had done was he was growing the bacteria. You can see all the little colonies of Staph aureus, and then we see that up here there's this big mold colony that contaminated his plates. Fleming may not have been very careful to keep the lids of his Petri plates closed, and so here he had seen the penicillium mold colony, and the first few times he looked at that and just threw it in the trash because he didn't want to study mold. He wanted to study staph bacteria. But then, at one point, it's reported that he looked into the wastebasket, and said, something weird is going on. Not just that mold has contaminated his plate, but wherever the mold colony had grown, the staph bacteria were inhibited from growing. He noticed that it was like there was this force field or this zone where the staph bacteria were prevented from growing. And he concluded that there must be some sort of chemical that is getting into the environment from the mold colony that stops the staph bacteria from growing. And that's how penicillium was discovered. Penicillin, the antibiotic. The mold is called penicillium, and when you look at these molds, as they grow, little droplets of liquid will come out of the penicillium colonies. And so penicillin was actually fairly easy to discover once this realization, this connection had been made, because we're going to grow penicillium in lab, and the penicillin antibiotic will come oozing right out of these colonies. And so you'll see that later on in the course. And in fact, Fleming's discovery didn't really get implemented in a commercially available, broadly applied sense until the 1940s. There were two scientists by the name of Florey and Chain, and Florey and Chain were the ones in the 40s that finally got penicillin into mass production, and that saved countless lives, soldiers, especially during World War II. And the rest is history since then. All the different derivatives of penicillin. We have, as I mentioned, methicillin, and there are many other cillins. They all end with that suffix. You've got tisseracillin and oxacillin and cloxacillin and piperacillin. We could keep going. So you'll hear of many penicillin derivatives that started from this event here, and antibiotics after that point. Now, we do have a review slide at the very end, and I'd like to also save this for our Zoom discussion, just to make sure you guys are on the right track and that you're making the connection between the scientists and their contributions. So I'll leave it at that, and that concludes day 2 of lecture 1. And next time I see you guys, we'll be on to lecture 2.